I need to emphasize in the very beginning that my experience comes mostly on humans. I have done some rodent studies in my early career with sugar rats, and then actually we gave them beta-3 agonists, so kind of I came back to this area after many years. But yes, originally I come from Turku, but not because of the many roadworks. This time I came from Sherbrooke. So I just started there as a visiting scientist, so that's what I will do for the next year. About brown adipose tissue, as you previously in the previous presentation heard about the storage part in our body, this will concentrate on the dissipation part then. There are a few points that I wish to make before I'm going to the effects of obesity in brown adipose tissue. First of all, in newborns and in rodents, we can find brown adipose tissue in the subscapular region. It's similar as not exactly, but almost exactly the same region which can be found from rodents. And in adults, I hope that this video will, no. Can I ask help? Can you push the, oh, excellent, thank you very much. In adults, the main depot can be found from the supraclavicular region, which is found in the neck area, just a couple of centimeters inwards from here. And that's the main depot. But other depots exist as well, like you can see from this image, which is a FDG image reflecting glucose uptake in this particular lady. So the bright areas reflect the highest glucose uptake, highest metabolic activity in the body. And the location is one point which probably differs adult brown adipose tissue function from rodent and infant function. And the size of the tissue is also one point. When we are born, we have 1 to 5% of our body weight is brown adipose tissue. And it actually remains, it means approximately 100 to 200 grams of tissue per body. And that remains quite same during our adult life. So at maximum, we commonly find 100 to 200 grams of brown adipose tissue in human body. But you understand the contribution in a bigger body size is different than in newborns. Another issue is that the brown adipocytes we have, they are two types of adipocytes. They are so-called beige or bright adipocytes or the classical brown adipocytes. The classical brown adipocytes are especially the ones which are found in rodents and in newborns. And they are typically including very small lipid droplets and a number of mitochondria inside the cell. The beige adipocytes may be described like this. They are kind of prepared for either transferring to brown adipocytes in times of need, like with chronic cold, or then turn to white adipocyte type cells in times of caloric excess. But in the depots we have, it is always a mixture of all these three types of adipocytes. So investigating or doing research to the specific depot which we can find, you can't say very exactly what type of adipocytes you have there. But in rats, there is a study already from 1990s which suggests that the inner parts of the brown depot is the classical type, cell type. They also, the classical types include less lipid, as also can be demonstrated with these images. And the external layer, which is uppermost to the skin, it's including more lipids and is more like a white adipocyte type. And the last issue or point is thermogenesis. Cold is very potent activator of brown adipose tissue function. And in rats, when they are put to cold, they transdifferentiate almost all adipose tissue which is in the body to brown. While in calorie excess, they transdifferentiate even the existing brown sites to white. But obviously, this cold-induced thermogenesis, it increases energy expenditure significantly in rodents, but also in humans. This is a study from Japan, from Yoneshiro and Saito. They measured a cold-induced thermogenesis, which means that they measured energy expenditure in cold and in warm, and the difference is the cold-induced thermogenesis. And there is seasonal variation. So in winter, this increase is much higher. It means in calories, approximately in PAT-positive subjects, this means like 200 calories per day. It's quite much. But in persons who don't have any obvious or detectable brown fat, the increase in winter is approximately 20 calories per day. That's not neither much, but I'll come back to those a bit later. So maybe in humans, the thermogenic potency is not that high as in rodents, as can be illustrated by these images. The heat or the therm generated by brown adipose tissue may be detected by infrared camera. And the skin area only in here is increasing by cold activation. So the kind of the heat produced for whole body is not that obvious in humans than it's probably in rodents. One of the most important protein for heat production is UCP1, uncoupling protein 1, which is able to uncouple the respiration chain in mitochondria and produce heat instead of ATP. There are animals which don't express UCP1. However, when, for example, pigs are put to cold, they start to generate heat by some other mechanism, maybe or ATP synthesis is one of the suggested mechanisms. So these are kind of the basic things that I wish you to keep in mind during my following talk, that there might be some of these differences between humans and rodents. Cold is increasing metabolic, overall metabolic, it activates metabolism in brown adipose tissue. It increases glucose uptake rate in brown fat. It increases blood flow in brown fat. It increases oxygen consumption in brown fat, and also fatty acid uptake in brown fat in lean, healthy humans. And as the oxygen consumption increases in brown fat, it doesn't do so much in other tissues like muscle and white adipose tissue. It has been regarded as an important contributor to thermogenesis in humans as well. However, when we investigated the other inner or deeper muscles as well, we found that actually there are those deep muscles which increase in oxygen consumption significantly during cold, which may mean that brown fat is a minor and probably the muscles, deep muscles, the more significant contributors to cold-induced thermogenesis. This doesn't mean that brown fat is not important. We think that brown fat is the first and the most important trigger for the increased oxygen consumption in the deep muscles. And even more, the importance of brown fat daily energy expenditure is clearly correlated to blood flow perfusion of the tissue. So it must have an important role in our, at least in triggering thermogenesis in our whole body. What about in obesity? There is also already some evidence showing that probability to detect brown fat activity is inversely related to body mass index. And it is well known that brown adipose tissue function is impaired in these obese rodents, and further that transgenic mice which have decreased brown fat, they easily develop obesity. And of course the question is like this: these brown adipocytes become more like white adipocytes in appearance and function. So having said that, maybe you can call increased lipid droplet size in brown adipose tissue a ectopic fat accumulation. So in humans, glucose uptake is clearly decreased in cold in obese subjects when we compare them to lean. This is what we most commonly found in PET imaging. This is FDG imaging. Lean subjects, 70% of them show this kind of accumulation of FDG as a signal of increased metabolic rate in brown adipose tissue, while our obese subjects most commonly look like this. So they don't have any activation in spite of a similar kind of cold exposure. Also, what we can find in obese subjects is that insulin-induced glucose uptake is reduced, showing that brown adipose tissue becomes easily insulin-resistant in obese subjects. And insulin-stimulated and cold-induced glucose uptake, they go hand in hand in obesity and also in lean subjects. Not only glucose uptake, which is important, of course, but it is regarded that brown adipose tissue anyway utilizes more fatty acids than it does glucose. Cold exposure increases fatty acid uptake in brown fat fourfold if we compare these PAT-positive subjects to PAT-negative subjects. And if we compare them to obese subjects, the increases or the difference is even higher. This difference between lean and obese subjects can be found already in room temperature, so there is no need necessarily to expose the subjects for cold to find whether one is PAT positive or not, based on imaging. As the Adipocytes in brown fat or beige fat region have accumulated more fat. The higher lipid content impairs the oxidative capacity. So metabolic rate of oxygen is closely related to the triglyceride content in the tissue. This CT radiodensity is based on a computed tomography where the tissue density, the lower or the higher is the radiodensity, the Hounsfield units, the less there is triglycerides. So these are the fatty brown adipocytes, and these are less fatty adipocytes. And also this metabolic rate of oxygen in brown fat is inversely related to body mass index, again suggesting the correlation between obesity and brown fat functional capacity. So overall metabolic function is decreased in obesity, and blood flow as such is decreased as well. But this increase by cold can be seen still in subjects. If you are lean and you don't have detectable brown adipose tissue, you still may be able to increase your blood flow in brown fat area. Along with these metabolic changes in brown adipose tissue, we can find some changes in insulin concentrations and fatty acid concentrations in systemic blood circulation. So insulin levels go down whether you are obese or lean, if you are exposed to cold. And fatty acids go up nevertheless you are lean or obese, but it does so to a lesser extent than in lean subjects. These lean subjects who are PAT positive, they are able to increase their fatty acid concentrations even more, so reflecting the lipolytic capacity by the activation of sympathetic nervous system in cold. However, energy expenditure, whether you are obese or lean, again is capable of increase during cold. That's kind of, in the very beginning it was against our expectations, but this can be found by several groups which have measured energy expenditure during the cold exposure. What if we then put our subjects to, or introduce our subjects to low-calorie diet? After 5 months, when they have lost more than 10% of their original weight, they are able to increase cold-induced glucose uptake in brown fat. Similar kind of finding was also shown by Virtanen and Van Marken-Lichtenbelt from Maastricht. Gastric banding bariatric surgery 1 year after surgery, when the subjects or the patients lost more than almost 30% of their weight, they were able to increase PAT metabolic activity significantly. What does it actually mean if one has active brown fat? How much energy you are able to consume? This is based on our cold exposed studies, and assumptions are made on approximately 10% calories come from glucose and the rest from fatty acids. And the first one is male who likes to swim in at winter time, and he had 60 grams of brown adipose tissue in supraclavicular region. And converting that to energy which can be burned from white adipose tissue it was equal to approximately 3.4 kilograms in a year. That is not much if you have a patient with 50 kilos of overweight. So you have to take like 10 years with this kind of activation. But also in a lean subject a female who had only 10 grams of brown adipose tissue, she was able to burn approximately 1 kilogram of white adipose tissue based on this activation. Of course, based on oxidative metabolism, it is estimated that 15 to 25 kilocalories per day is the amount which a brown fat can optimally burn. But again, when converted to white fat, it's approximately 1 kilogram. I think this is not much, but it's important for the lean subjects, as they are able to maintain their body weight, probably partly because they are burning 1 kilogram per year. But for obese subjects, that may be too challenging. Are there any other than this cold? This cold acclimation and exposure has been shown already in lean subjects quite many times, so that obviously works, but there is no studies available yet for obese subjects. Whether it works or is applicable for our obese subjects is not known yet, or whether there is some other means which needs to be used before cold acclimation. These might include, for example, capsinoids. These are just examples of what kind of data is available. Again, cold-induced thermogenesis increased with capsinoids with 6 weeks of treatment. And of course, the very promising drug, which is expected to have exceptional effect on brown fat, as we know that in rodents it markedly increases energy expenditure and decreases obesity in rodents. In one dose, 200 milligrams dose, lean males were able to increase their calorie consumption by, or not calorie consumption, but energy expenditure by 200 calories per day, which is significant. More studies are coming around this, and I'm sure that a number of groups are working with this at the moment. So, of course, you know, diet, exercise, cold, whatever might be good for improving health and metabolic health, and also that exercise has been suggested to improve brown fat function. It has actually quite similar effects than cold to our systemic substrate concentrations, and irisin was one of the substances which was suggested to have improvement in brown fat function. It was introduced by Boström and Spiegelman that induced, when rats were induced to exercise, it stimulated browning and UCP1 expression in subcutaneous adipose in mice, and this was based on 10-week endurance exercise. This is very debatable subject at the moment. There are reports like this that suggest that irisin plays a role, and there are reports that suggest that there is no role for irisin in humans. So that will be resolved in, hopefully, in becoming years, whether or not it has any importance in our metabolic health and brown fat function. We did some studies exercise studies in Turku, where we had high intensity training and moderate intensity training groups which obviously were able to increase their metabolic activity, glucose uptake in skeletal muscle, but not in brown adipose tissue. However, when we selected the 6 subjects which were brown adipose positive in the beginning of the study, they actually decreased brown fat metabolic activity by the exercise. And we also repeated this kind of study with longer term exercise training, where we found no effect on brown fat function whatsoever. So maybe there is more than thermogenesis and energy expenditure and glucose uptake in brown fat. And it's well known that brown fat has the capacity of burning fat. The primary site is intracellular lipid droplets, but it also may deplete those intracellular lipids by taking up fatty acids from circulation. And this might optimally protect other tissues from lipid load, like, for example, brain. And cold, again, was shown to increase brown fat radiodensity, which means that it burns the intracellular lipids. The lipid content decreases by cold. And this is, again, found both in brown fat negative and brown fat positive subjects. And this, as already I mentioned, this oxygen uptake rate is related to triglyceride content in brown adipocytes, which is also related to fatty acid uptake, suggesting that the depletion really takes place. So animal studies have shown that this activity, brown fat activity, might control triglyceride clearance. They showed this by exposing rats for cold, and showed that there was a difference in triglyceride elimination. Further, the same group showed that brown fat activation reduces hypercholesterolemia in the mice if they are if brown fat is transported to them. And we measured a bit similar kind of correlation that when fat fraction measured by MR is in cold, it's related to cholesterol concentrations. But also glucose homeostasis and insulin sensitivity has been shown to improve in animals. Again, brown fat was transplanted to mice, and it really showed clearly that both glucose levels and the area under curve during glucose tolerance test was improved by the transplantation. We measured the triglyceride content in brown fat and compared that to insulin sensitivity measured by clamp, and found that brown fat lipid or triglyceride content is inversely related to whole body glucose, whole body insulin sensitivity. This I will skip. As a summary, brown adipose tissue function is impaired in obesity, both in rodents and in humans. And I may suggest that thermogenesis may be more important in rodents for the whole body perspective, while in adults it's mostly local. And brown fat function may be improved by cold, weight reduction and drugs, but definitive results about drug treatment are still missing. These are the people who are who have worked a lot for these data, which I've shown from our lab, and I thank you for your attention.
I had two, but I'll ask one. I found that incredibly interesting. I just wanted to ask about the exercise intervention that you used in terms of looking at brown adipose tissue. Did it differ to previous groups that have looked at exercise? Because I'm wondering whether it's to do with the type of exercise that you're getting them to do and whether that has more of an impact, whether it's resistance training compared to, I don't know, more cardiovascular stuff.
So the type, there was no differences with whether it was high intensity or moderate intensity type of exercise. And the longer term was more like moderate type of exercise, so we didn't, in that study, determine.
If they were doing resistance training, which kind of encourages muscle growth, whereas when you've got something like running, that's more, sorry.
It's more like endurance training.
Yeah. All right.
Like gym. The long term was supposed to be such that it's kind of mixed, so it's not concentrated on any particular type of exercise, but combining both endurance and more aerobic and every.
Yeah. All right. Okay. Thank you. Which was the best?
Yeah, thank you very much for this talk. I also just had a question about what does cold actually mean in human studies? How long, what temperature are you talking about? And especially regarding to the number you said, the 25 calories per day that can be kind of lost through this. Does that mean daily? Yeah, what does that actually mean in terms of how the cold gets to the person in a way?
The exposures we used is kind of mild. So what we used, we had two types of cold exposures. The first one is we exposed the subjects for cold air, 19 to 20 degrees or 17 to 19 degrees for a couple of hours, and then we put the food into cold water during the scannings. And the other one is such that the subjects are lying between the sheets. With the sheets have perfusible water inside, so they are lying in between of cold blankets, and they stay there for 2 hours, and that's more like individualized, so that we start from room temperature and then we lower until they feel shivering, and then we increase the temperature a bit.
So in the terms of the 25 calories, would that mean they would have to do that every day, or does that mean that it overall changes? I don't know.
Yeah, it's
looking
kind
at
of,
me and
yeah.
laughing. Is it a
Per
stupid
day,
question?
it's per day, but there are those cold acclimation studies which are published so far. There are from 2 to 6 hours acclimation per day, the length which has been used.
Thank you.